I would like to express my deep gratitude and thanks to GAF. At the same time, I would like to also thank Dr. Damle for nominating me to give a lecture in this plenary session on this topic, which is very close to my heart. And this is a traditional medicine which used to be practiced by my ancestors. And we were living in a village where snake bite victims used to be there once in a month at least, because it is surrounded by estates. And the primary health center did not have any medication required for these snake bite victims. And they were dependent on our family. And this medicine has been, in fact, traditionally practiced in our family, which I'll be talking about it in few minutes. And because you know that the snake bite is a most neglected disease in the medical field. Why? Because it has become very common. If somebody gets bitten by snake and dies, and sometimes the treatment is available, medical facility is available nearby, and sometimes the medical facility is not available. But it is very, very devastating for a family which is very poor and dependent on a bread earner who passes away because of the snake bite. Sometimes they don't respond even to the traditional medicine that is available, that is especially the horse antivenom. And this is the statistics that I've got recently from the Hindu newspaper. Every year there will be about at least 35 to 50,000 mortalities because of the snake bite in different parts of our country, and about 140,000 disabilities which are associated with the comorbidities of the snake bite. And the prominent snakes, all of us know, they are called as big four: cobra, Brussels viper, crate, and saw scale viper. And whatever the medicine that has been practiced till date, some traditional medicines are used, and they have saved the lives of people. And wherever the medical assistance is available, the horse antivenom has been administered. But there are many complications associated with the horse antivenom. One, it needs the medical assistance, the medical practitioner or a nurse to administer it intravenously. Second, it needs to be stored in a refrigerated facility. Otherwise, it loses its activity. It undergoes denaturation. And many remote places in India, villages, where there is no primary health center available, plus even the electricity supply may not be there, and refrigeration facility not available, the storing of horse antivenom becomes very difficult. So therefore, such victims who are in the remote areas, working in different remote fields, sometimes when they get bitten, it is too late for the medical assistance for them. And before they reach the medical center, they might pass away. Then the horse antivenom, whenever it is administered, everyone knows that it does cause anaphylactic reactions and other comorbidities. Similarly, at the same time, sometimes the horse antivenom is in short supply. It is not available because it is not supplied to that center. So that becomes a very difficult situation for the victim. And the medical center may not be there in nearby area. So therefore, another most important aspect is about the cost of the horse antivenom. It is very costly and a poor man cannot afford it. Therefore, the Ayurvedic treatment through the herbal remedies assumes significance, at least in the form of first aid to save the life of the person. There are many, many documents available in the literature, and most of them have worked, and many scientists have worked in India on different types of the herbal remedies on the snake venom, carried out research on the snake venom of different species of the snakes available. The predominant of them are just listed, but it doesn't mean that this is the references that are available. There are many more references are available. One of the prominent one is Andrographis paniculata. A lot of work has been done on this plant to study it as an antivenom property on Naja, Davoyorasali venom, and other that is saw scale viper venom. Similarly, the hydrocotyl javanica and gloriosa superba. So then Pentese burmanica is another plant which has been worked on Naja Kautia venom and prevents lethality. Hemidesmus indicus, also known to prevent lethality and hemorrhagic activity of viper. All these are experimental evidences on the animal studies and not on human studies. And maybe some tribals might have been using these medicines, and they have been practicing it to treat the victims of the snake bite, and it has worked in the traditional medicine in the tribal belt, tribal areas. So going to my topic on which I have worked, the plant material, that is Coex lacryma jobai, the root extract. This was our family medicine, and this used to be given to the snake bite victim by my ancestors, and after that of my mother, it stopped because we are all urbanized now. We don't practice this Ayurvedic medicine, but it remained very close to my heart to carry out some research on this. And one of my PhD student, Rajesh, worked on this. And we did find some good results as far as some of the enzyme activity inhibition, in vitro enzyme activity inhibitions are concerned, like hyaluronidase and L-amino oxidase. And these are the papers that we have published from the work carried out using Coex lacryma jobai ethanolic root extract. And we found that the ethanolic root extract is not toxic in the rat or mice at the dose of 2 grams per kg. And we also carried out some necrosis and hemorrhagic, anti-hemorrhagic activity with the root extract at a dose-dependent manner, that is 100 milligrams per kg, 200 milligrams, and 400 milligrams per kg. And you may see to the extreme left of yours the control where the animal skin at the dermal injection was given. 30 microgram of Russell venom was given, and at the same time it was treated with the standard horse antivenom. And you may see the decrease in the size of that hemorrhage only a little bit, but with the treatment of the ethanolic extract, which was administered, okay, orally to the animals, showed significant results and decrease in hemorrhagic activity. And we did carry out some of the in vitro venom enzyme inhibition studies, and we did find good results with venom enzymes like hyaluronidase, phospholipase A, and protease. For hyaluronidase, we used hyaluronic acid, which is available commercially, and phospholipase A, we used human RBCs, which were washed in the physiological saline to find out the hemolysis of RBCs. And for protease, casein hydrolysis activity was carried out using standard procedures of these references. And all 4 snake venoms we tried for the antivenom activity with the Coex lacryma jobai extracts. We used 4 different types of extracts. First, dried root ethanolic extract at 100 microgram and 200 microgram per kg level, and fresh root ethanolic extract, that is just fresh root taken from the plant and macerated in ethanol at 100% maceration, and 100 microliters and 200 microliters we used for the test. It is all in vitro experiments, not in the animal studies. I'll come to it a little later. And we also tried lyophilized aqueous extract, which is stabilized with 2% starch. And I'll come to the point why we had to stabilize with the 2% starch a little later. And the fresh root aqueous extract. And you may know that fresh root aqueous extract will be useful only at the site or very close to the place where the victim is bitten by the snake. But it is not stable and you cannot keep it in the laboratory temperature, the fresh root extract for long duration. But only for the experimental purpose, we have used the fresh root extract. Now we have the results here. So 50 microgram of Naja venom, hyaluronidase activity was studied, and 100 microgram of hyaluronic acid was used. And you see here the results. In fact, the dose-dependent manner inhibition of the activity was observed, and the activity with dried root ethanolic extract was found to be higher than the fresh root ethanolic extract or the lyophilized fresh root extract or the fresh root extract. Similarly, we carried out work with Russell venom And we found a similar activity up to 40% inhibition with the dose-dependent manner. And with the Echis carinatus venom hyaluronidase activity, the dose-dependent manner inhibition was observed, and with Bungarus cerulus also. Then we carried out work with phospholipase A activity, where human RBC, 100 microliters of human RBC was used, and 50 microgram of Naja venom was added. Before that, the root extract was added to it, and inhibitory activity was studied. And we measured the hemolysis of RBC using the U visible spectrophotometer, and the percentage inhibition was calculated. And you see the activity here, quite good activity was found in all the 4 different types of extracts with Naja venom, Daboyarasali venom, and Echis carinatus, and Bungarus cerulus. Then we carried out protease activity, where casein, 2% casein hydrolysis study was carried out, once again using the U visible spectrophotometer, and percentage inhibition was found. And there was good activity with dried root ethanolic extract and fresh root alcoholic extract for Naja venom protease activity. Activity. The few differences are there. In some, the fresh root is showing good activity, and some dried root is showing activity. But most important point is there is inhibition activity in the root extract. This is Echis carinata protease activity inhibition, and Bungarus cerulus. Then we did carry out the fibrinolytic, that is inhibition of fibrinolytic activity per kg ethanolic extract for the Daboyarasali venom as well as Naja venom, and there was significant inhibition. I have not projected the graph. Similarly, 100 microgram ethanolic extract was also capable of showing nucleus inhibition activity of Naja venom. And some activities carried out in animals where with the Daboyarasali venom, where hemorrhagic activity was neutralized with 400 microgram of ethanolic extract, and 30 microgram of the Daboyarasali venom carried necrotic activity was inhibited by 400 microgram of ethanolic extract. Then 2 microgram per kg Daboyarasali venom and 4 microgram per kg Naja venom edema was inhibited by 400 microgram per kg ethanolic extract. Then coming to the actual in vivo studies, that is with the mice, we carried out a lot of other preliminary studies of LD100 dose for different venoms, but we could not do the work with other species because of lack of funds. We have done part of the work with Daboyarasali venom, LD100 dose for the intradermal, LD100 dose for subcutaneous, and LD100 for intramuscular. Here is the dose that is mentioned, 8.5 microgram per gram venom of LD100 for intradermal, and 10 ml per kg aqueous fresh root extract was given in 3 doses at hourly interval, and 6 animals, there was no mortality. Similarly, subcutaneous LD100 dose was 9.8 microgram per gram, and fresh root extract was 10 ml per kg, and 3 doses hourly interval, no mortality. Intramuscular, 8.5 microgram venom, and similarly, there was no mortality. Then coming to the other type of extract, here we treated the animals with the LD100 dose, that is injection, intradermal, subcutaneous, and intramuscular, and fresh root extract, aqueous ethanol extract. Sorry, fresh root ethanolic extract was administered, and it was diluted 1:5, and the extract was 50%. That is 50%. That is 50 grams of ethanol. 50 grams of root was macerated in the 100 ml of ethanol, and that was diluted 1:5 and given orally. And see, 4 animals were treated in each group, that is intradermal, subcutaneous, and intramuscular at LD100 dose, and there was no mortality. Then the lyophilized extract. Why we had to carry out this work? Because I wanted to see if the root extract can be lyophilized and used as a stored material. And if it could be transported to the remote areas where it can be kept in the laboratory temperature and utilized by a person as a first aid measure. I wanted to do the study. Therefore, I prepared a lyophilized fresh fruit extract. When we first prepared plain lyophilized fresh fruit extract, we did not get good protection. There was more than 50% mortality. Then one of my scientist colleague from Pennsylvania University told me that some of the active ingredients of the root extract may get denatured when you lyophilize it. It is better you use dextran or starch or any other stabilizing material, which tricosan or something like that, which will stabilize the material. So I use simple starch, 2%, and lyophilized root extract was used, fresh root extract, which is aqueous. And we found with the LD100 dose, when 500 milligram per kg, it is powder, which is given at a dose of 500 milligram per kg, and 3 doses were given orally, and it was found that there was 100% protection against the W. venom. So my contention is this preparation of the coax lacrymojobi, if it could be used as a first aid measure to prevent the death of the individuals, it would be worthwhile. Here, I'm talking about stability of the extract. We found that the ethanolic root extract, fresh root extract, prepared and kept in the laboratory temperature for 1 year showed the same effect, same activity, and 100% protection in the W. venom, LD100 dose. Same way, lyophilized root extract kept in the laboratory temperature for 1 year also showed 100% protection against W. venom. So my contention is there is no need of fridge or there is no need of freezing facility. The root extract in the ethanol or in the lyophilized form is stable even after 1 year, and it could be used as a first aid measure for the persons who are bitten by the poisonous snake. It could be possible that we have to carry out some more work with the Naja and Echis carinata, Bungarus cerulus. Possibly we may get same type of results. Due to lack of funds, I could not carry out much of the work in these other species. So we have seen that there is 100% protection against W. venom at LD100 dose intramuscular injection of W. venom to the mice, as well as intradermal or subcutaneous. And I still feel that it can be used as a first aid measure. And I would like to give my acknowledgement and thanks to my PhD student, Rajesh, who has worked extensively on this topic. And I would like to thank Dr. Murali, Deputy Director, New Care, Nitte University, who helped me with the animal experimental studies. I also would like to thank Dr. Rishikesh Damle, who helped me and encouraged me to move forward with this work. And he's my, in fact, I should say that my close partner. And I would like to come out with my product with him. And I would like to thank Birac Pace for giving me partial grant, and only the first installment grant they have given me. And I would like to thank Irula Snake Catchers Cooperative Society for giving me the venom. Thank you very much. If there are questions, I would like to answer those. Thank you.
Can you guys hear me?
Yes, sir. Yes.
Yeah. Thank you very much for that enlightening work, and I've seen through this work you are doing since many years, and I know that you have also done further work on purified molecules from these plants. Anyway, probably you have your own reservations on sharing the data. That's fine. I want to ask you first question. Meanwhile, subsequently others can ask the questions. First question is, what was your idea when you went on doing these enzymatic assays?
Yes, sir. First of all, we wanted to see whether actually this works on enzymes of the snake venom. Those are the major toxins that are found in the snake venom. And in vitro, if we can prove that these are getting inhibited, then we can carry out the work with in vivo studies. And possibly that must be the mechanism which will work in human beings also. where the enzymes of the snake venom are getting inhibited when this preparation is administered orally to the victims.
Okay.